Thanks to the organizers. So I'm going to talk about DNA sequence, how it governs the capillary forces through protein DNA co-condensation. But before I get into all these terminologies, let me discuss how the DNA is organized within the nucleus. So to get the perspective of what we are actually looking at, if we compare the DNA, which is double in length compared to the distance between Earth and the Sun, and compact it in a tennis ball, we would reach to the similar level of compaction as we are looking into the DNA which is present in the nucleus. Now, this is not the most amazing part of the story. Now, what happens is that this DNA can reorganize itself inside the nucleus depending on which genes wants to express. And there are several mechanisms which actually play an important role to reorganize this nucleus, right? So one of the mechanism is formation of these co-condensates. So what we are looking at right there is the DNA, and the proteins come together and they form these droplet-like structures, which basically helps regulate genes, right? So we would be talking about these tiny droplet-like structures. So people have seen that in many experiments that when they introduce these proteins along with the DNA, they form these vesicle-like structures. And here the experimental setup is like that they have fixed the two ends of the DNA, and then when they introduce the proteins, they see these condensates. In the next set of experiments, what they are doing is that they are looking at another protein, which is HP1, and what HP1 does is that it reorganizes the DNA inside the nucleus. And when it is in presence of the DNA, you can see that these tiny droplets are formed when these proteins interact with the DNA and form these droplet-like structures. Here, the experimental setup is a bit different, where you have these two optical beads, and in experiments, one can control the length between these beads, so that controlling the amount of DNA which can be present inside the condensates. Next, we also see that these condensates can actually apply force on the DNA such that and this force is at that level that this DNA actually breaks. So what we understand up until now is that the DNA is present inside the condensate in the compacted form, and there is this force that this condensate applies on these bare DNA parts, or DNA which is out of the condensate. Now, as we know that DNA is inhomogeneous in nature, what it means is that there are these different nucleotides which are arranged on the DNA. And so the key question we ask is that what is the role of the DNA sequence in co-condensation? So we start with a very simple model where we have these green dots, which are basically proteins, and this blue thread-like structure which models the DNA. Now, in a homogeneous case, we have these different regions of the DNA which interact with equal strength to the proteins, whereas in the next model we have these two regions. One is shown in red and the other one is shown in yellow. Now the red one interacts strongly with the proteins, but the yellow one interacts weakly with the protein. So when we simulate it, we find out that in homogeneous case, the condensate is formed near to one of the ends, right? But in the case of heterogeneous, where the strong interaction is in the middle, they always form right in the middle. So for a comparison to the experiments, this is the recent paper which has been submitted to bioRxiv last Saturday, and what they are doing here is that they are looking at this DPS2, which forms co-condensate in the presence of DNA. And this DPS2 has a specific property that it doesn't differentiate between the different sequence on the DNA, and they found out that these condensates do form at one of the tethered end, right here or right here. So for another comparison, for the heterogeneous DNA, we have our collaborators from IIC who do these experiments of DNA and LSR2 protein which binds to the AT sequence on the DNA. And we found out that in purple is shown the AT content, and as you can see, where the AT content is higher, the condensate is forming right there, just like our model here, where the red regions which have higher interaction with the proteins get compact in the co-condensates. Next, we wanted to know what regulates the force on this bare part of the DNA, or the DNA which is outside the condensate. So to model this, what we did is that we tuned a particular parameter, which is basically the end-to-end distance, and by increasing the end-to-end distance, we make sure that the amount of DNA which is inside this condensate gets lesser and lesser as we increase the end-to-end distance. And we calculate the force which is exerted on this bare DNA. So what we found out is that for a homogeneous DNA which has equal affinity for the proteins all over, we don't see much increase in the forces. But next we look at the heterogeneous DNA, which is shown in purple here. For lower end-to-end distance, it has lower force, but suddenly the forces increase. But exactly what is happening is shown here, that when you have lower end-to-end distance, your weak regions, which are shown in yellow here, forms the interface. But when you increase the end-to-end distance, you are basically stretching the DNA so that the red regions, which were earlier inside the condensate, are now coming out on the surface. And that is why, because they have higher interaction with the protein, they are exerting more force. So to conclude my slides, the position of the condensate is determined by sequence heterogeneity, whereas the forces on the bare DNA, or the DNA which is out of the condensate, is determined by interfacial DNA motifs. So I want to acknowledge Sandeep, Pinaki for their guidance, my collaborators Tejas, Prakshi and Mahipal, and IMSC Computational Biology Group and HPC. Thank you.
Any question?
Do you have a measure of this heterogeneity? I mean, like, can you give a quantitative metric of the heterogeneity and see how that correlates with the amount of condensation? Do you have any such curve?
Yeah, so we have done such experiments where we do not consider this particular model right there. We increase the heterogeneity by introducing this piece of DNA. Specifically, we are introducing lambda DNA, and then we see where the condensates form. But due to less time, I'm not presenting those slides.
So what is your metric there, the heterogeneity metric?
So the heterogeneity metric is basically in terms of KBTs that we use in Lennard-Jones potential, the epsilon of Lennard-Jones potential. And what we do is that we look at the AT content in 10 base pairs, and based on the AT content, we decide on the strength, interaction strength.
Short question. Is there any relation of this condensation with what is there in a chromosome?
Yeah.
Where you have DNA wound on histones which are hierarchically organized?
Yeah, so while we look at the nucleus, there are different levels of organization. For example, DNA can be organized in form of nucleosomes and even higher level of organization, but DNA sometimes are without any proteins like histones, for example. For example, between two nucleosomes, you can find a protein which is not interacting with any of the histones, right? So you get this bare DNA, and then what happens is that these proteins, which are very important for gene regulation, especially during the development, they come in and they bind to those parts of the DNA, and such that these different DNAs parts can come together and help regulate genes.
Thank you.
Thanks.
Yeah, so I wanted to ask, let's say if you include these laminar interactions in your model, like with some energy scale, so does it then prevent this coalescence of the droplets that you are seeing?
Yes, it does. So in that case, as I was showing in the experiments, that we do see that there are these multiple condensates that form on the DNA. And the basic reason behind formation of these multiple condensates, we think is the DNA sequence. And when we simulate our lambda DNA model, which is not shown here, we do find that there are these multiple droplets that form on the DNA.
You don't have any laminar interactions, right, in your model?
What do you, sorry, I didn't get it. What do
So
you mean by
like
that?
you have like just DNA protein interactions in this case. So what I'm saying is you will also have basically in the chromosome, you will also have interactions with the lamina. So if you include that in your model, because in your simulations you
right.
saw that two of the droplets coming together and forming one.
Yes.
What I'm asking is, will that actually oppose that if you include it?
This one you are talking about, right?
Yeah.
Yeah, so we are looking at a very small fragment of DNA. In fact, its length is around 0 to 5 kb. So it's not a very long DNA, such that we don't include the effect of histones as well as the other parts of the nucleus.
Okay, we can discuss about this more.
Yeah.
Okay, so let's thanks Rohit again for his nice and short talk.